Allison Cleary and Ludmil Alexandrov, it's such a pleasure having you here at SciLifeLab and celebrating your Science and SciLifeLab Prize. Congratulations.
Thank you.
Thank you so much.
So today I thought that we would take an opportunity to talk a little science. And I'll start with Allison, the grand prize winner. And I just wanted to ask you to describe a little bit on your cell corporation.
Sure. Really, the work that we did was based on this observation, really, that there are, within an individual tumor, there are these multiple subclonal populations of tumor cells. And because of the way we traditionally think about the clonal evolution of tumors, we traditionally view these subclonal populations as being sort of self-interested competitors with respect to the way the tumor is organized. But we were interested in this idea of, instead of competing with each other, if maybe these different cell populations might
Okay, so that's an interesting, you said
each other.
we were interested in, you say that you just were interested in, why they are not competing, why they are working together. So what made you think that?
Well, I mean, if we think about the evolution of tumor cells in the context of Darwinian evolution, we know, looking at the different species that have evolved presumably from a single species, in nature we see all kinds of different interactions between all of those different species, not just competitive interactions, and we thought there really shouldn't be any reason why those different types of interactions can't occur between the cells within the epithelial compartment of the tumor. Certainly we know that the epithelial tumor cells interact very heavily with the surrounding stromal cells, with intervening immune cells, and so we thought that those similar interactions could be occurring between the cells in the epithelial.
This is very thought-provocative, because the original view is that cancers are like bacteria basically,
right,
and that they fight each other and they just want to grow, and that the growth comes from within, from mutations, that you have mutations in the cancer, in that cancer, and that will just grow. But you found that this growth is not necessarily. Can you tell us a little bit about that growth, and that the growth is maybe not just coming from within? Right.
So in the mouse model that we were studying is a model that depends upon a secreted signaling protein, Wnt1, which when it's overexpressed acts as an oncogene. And we found that in those models there's cooperating mutations in another oncogene, HRAS. But in a subset of the tumors, that mutation in HRAS was present with only in a subclonal population of the cells. And we were able to show that there was a cooperative interaction between the population of cells that was producing our secreted Wnt1 oncoprotein and the second population of cells that harbored that mutation in HRAS. And the tumors really depended upon both populations of cells in order for the tumors to grow.
Can you tell us a little bit, this is very exciting. You could see that you need, cancer cells to cooperate. I think this has huge implications.
I think it's a little bit scary, actually, and because, we think about this stepwise progression of cancer progression, you're accumulating these different mutations, and we assume that it has to be in the same cell that's accumulating these different mutations. But if you can have that stepwise progression, but those mutations are occurring in different cells that are then cooperating with each other, that changes the game, I think.
In what way?
Well, I think it makes it just so much more complicated, unfortunately. At this point, I don't think it's very clear whether or not these types of interactions are occurring in human tumors. I'd be surprised if they weren't at this point, but potentially it could be an artifact of using transgenic mouse models and expressing these different oncogenes in a large subset of cells as opposed to a single initiating cell. But I think it was really interesting questions.
So how does this implicate therapy, do you think? You know, because we want to get the treatments.
Absolutely. So for a situation where you might have a tumor that's dependent upon two or more cooperating subclonal interactions, I think we might be able to capitalize upon that and develop therapies that could interfere with those interactions or uncouple those cooperative interactions. And then
But if you look at today's therapy, do you think that they are limited by this cooperation?
I'm sorry, I don't understand.
Because today we also have therapies, and unfortunately, cancer is still a lethal disease for many.
Right.
Is the cooperation, has that anything to do with it, do you think?
Maybe. I think that regardless of the therapy, regardless of our approach, especially with targeted approaches, we're going to have trouble with tumors relapse and resistance developing, because if there's one thing I've learned, is that the tumors are really very, very good at getting around our interventions and getting what they need. So in our work, we were able to show that by eliminating this cooperative interaction, we got the tumors to regress, but actually, very interestingly, either of those two subclonal populations was capable of evolving independently of one another in order to rescue tumor growth. So the tumors, unfortunately, they find a way, and we're just going to have to try and anticipate what those steps are so we can be one step ahead.
We also have Ludmil here, and I think this is so, because Ludmil has looked at it from the mutation side, and you have looked at it from the cooperative side.
Right.
And sort of, we have in the history of, you know, how we thought how cancer has developed, we have been having two different type of thoughts. You know, it's an evolutionary process driven by cooperation, as you show, but also selection. And so this has been one viewpoint. And then there's been another viewpoint that it is driven by a mutator phenotype, that you have a high rate of mutations that have that are sort of driving the cancer. I interviewed also another prize winner, which was Thomas Lindahl, and he said, you know, probably both are a little bit right, both views. But it's so nice to have both of you here, because you're a little bit representing a little bit different viewpoints. You have tacked cancers from these little bit different viewpoints. So I want to also, you know, Ludmil, you know, how do you see when hearing about these cooperative cancer cells, how does mutations come into play here? Do you think that would these have different, because these are different clones, you can say, I mean, different cell types, and will they have different mutations driving them, or do you think that it's a more uniform way of generating mutations in the cancer?
I think this is a very good question, and it will be, so obviously I don't know the answer, but if I have to speculate, I think this will have different driver mutations. I think it will be interesting if we can look at that in human cancers. And I think yesterday we were talking with Allison. I think it's, unfortunately, we cannot just look from the genomic side. You really need to do a lot of experimental work to actually show that they're working in a, let's say, symbiotic way, or they're cooperating. But from the perspective whether one of them will be a mutator phenotype or whether it will be just driven by selection, again, from when it comes to clones, I am not quite sure what we will see, and it will be an interesting thing to explore. When it comes to human cancer, as you said, there's two hypotheses, and what we have been seeing in genomic studies, we have been definitely seeing the mutator phenotypes, but we have been
But
also,
not in all the cancers.
but not in all the cancers. So in
So
some,
how are they driven? I mean, the childhood cancers.
Exactly, exactly. Well, there is the argument of the bad luck of cancer. There is the argument. That you just have molecular clocks that are ticking with a constant rate, and they are in every single cell. And just by chance, they're going to hit KRAS or TP53, and you're going to get a cancer without having a mutated phenotype. And then there is obviously clear examples of mutated phenotypes.
Potentially, I mean, there's this, you need more than potentially one mutation in order to generate the cancer. And here, clearly, this is also cooperating between
Two different clones.
Different clones.
It's true. No, this is very, very true. But for childhood cancers, we have whole genome sequence childhood cancers that have less than 100 mutations.
Yes.
So it's definitely not a mutated phenotype. It could be some cooperation that's happening on the site. We haven't explored that, but I think there is definitely an element of selection.
So going a little bit to you, Ludmil, so when you have studied the reasons we get cancer, and you have been studying this you have basically been studying noise.
Pretty much, yes.
So you went for the noise, which is a very bold thing to do in science. And maybe you can tell us a little bit.
Yes, well, obviously we have driver mutations in cancer, and these are the ones that confirm clonal growth advantage, but we have a lot of mutations that are, as you said, noise. These are just the byproduct of the things that are mutating the cells. And in the majority of the times, people were throwing out these mutations, they were ignoring them. So what we wanted to do is we wanted to see whether we can actually look at the noise and whether we can actually split different types of noise or different patterns of noise. And there were studies from the late 80s and early 90s that gave us some indication that we may find something. And so, for example,
What was that?
So studies in the gene TP53, and they show that if you expose it, so they show that in skin cancers you have a very specific pattern of mutations. They showed in tobacco smokers that there is a specific, again, pattern of mutations. And then when you do the experiments, when you expose cells and when you sequence TP53, you see very similar things. So there was some indication, but the question is, can we deconvolute the noise and can we come? And we were able to, and we were able to find multiple processes.
So you, I mean, you're in the lab and we're scientists, and then you came up with a cocktail party.
Yes, that's true, yes.
It's,
yes, it's
something maybe unexpected from a scientist. Can you tell us about your cocktail party theory?
So it's the cocktail party. Yes, of course. So this comes. So when we were looking at the problem, when we were looking at the problem of mixing different signals or different noises, we looked at who has done this before, and we saw that the signal processing community has been trying to solve that. And the analogy was you're in a cocktail party and people were talking, and you have multiple microphones at different places, and you're recording it. And the question is, can you split out all the conversations? And can you see what different conversations are about? So there was a whole mathematical set of apparatus that was developed to do that. And we built upon it. And I don't think if this didn't exist, we were able to do it. So we took that, we modified it. It took several years to make it work. And then we applied it to cancer genomics. And then we looked at the genomes as microphones, that we looked at the processes that mutate DNA as the conversations. And then we try to separate them, and it's worked quite nicely, I think.
And we'll come back to that. But I also now want to bring in, Allison here, because what you see is that you have said that you have a cell, and within the cell you have different parties or discussions, or,
sure.
And that is contributing to the mutations. But I also want to challenge you. Then maybe you have cooperating cells, that you have different cells, and that you have one mutational signature occurring in one of the cell type and another one in another one, and that it's actually but you will take the tumor and sequence it, you will get the DNA from both. You're
That's
not
true. Yes.
identifying from the individual cancers. So what is your view? Do you think it's is it different processes occurring in different cooperating cells, or is it that there are three processes in one cell?
I think so. We have tried looking at subclonal analysis. So obviously, you take the bulk DNA, you sequence it, and if you sequence it deep enough, you can actually try to look at different subclones. And we have seen previously that different processes get activated, and sometimes we have seen that the subclone may have a unique process. But again, it's really hard to see whether there is cooperation there or whether there is competition. I'm not quite sure from genomics point of view how you can do it without doing the experiments Allison did with the mice.
Okay, so I also want to ask you about these mutational signatures, and because their essence, you want to understand the origin of cancer.
Yes.
And can you tell us about it? What did you study? What did you find? What is the origin of cancer?
So we wanted to, when you think about some cancer types, when you think about lung cancer, you immediately start thinking about tobacco smokers. When you think about skin cancer, you start thinking about exposure to ultraviolet light. But then the question comes, what causes breast cancer? What causes prostate cancer? And we wanted to try and go and look at, what's the origin, what causes this specific, all of the cancer types that we can get data for. And I think currently most of cancer researchers will agree that cancer is caused by somatic mutations, whether it's a cooperation or competition, but you need a generation of somatic mutations. So when we were saying that we studied the origins of cancer, what we meant is we wanted to see the processes that generate these somatic mutations. And to see that, each process may leave a unique fingerprint. So we were doing a bit of molecular CSI. We were lifting these molecular fingerprints from the cells. And we have them, and for some of them, we know the criminals that these belong, but for the majority of them, we really don't know. So it's a matter of doing the experimental work and doing the other side of the equation.
But you found some common sort of features between cancers. Tell us about it.
So there are some signatures that are very common. We found 2 signatures that we believe are molecular clocks that associate with actually age of diagnosis of patient. We found mutational processes that seem to be very common across human cancer, and this belong to the Appleback family of deaminases.
So those are the, So is there any other that were across?
We maybe not surprisingly, we looked at microsatellite unstable, and we all of a sudden, when we think about microsatellite unstable, we usually think about uterine, you think of colorectal, maybe stomach, but we saw across cancer, they are there, but they're at much lower percentages, so they're at 1 or 2% of breast cancer. And then pretty much we started finding things that are confound to a single cancer type. So for example, tobacco chewing will be very much confound to the mutational process of tobacco chewing will be confound to mouth cancers. You don't see it any other place. So there are some that are common, but they're not.
This is also interesting. How many would you say? Because when you have this exposure-generated cancers, the mutations are also prevented by a process called DNA repair.
That is true.
And this is often, for many of these type of lesions, it's a strand-biased repair, that you want to repair the actively transcribed gene. And so in a sense, if just to generalize, is it correct to say that the cancers caused by environmental agents are more transcription-biased mutations?
It's an excellent question. I mean, if we think that the majority of environmental caused ones, they cause bulky adducts that result in stalling of the replication machinery and recruitment of transcription capital, nucleotide excision repair. Yes, and I think there are many, many examples that we see that. And again, tobacco smoking, ultraviolet light, potentially some of the things that we eat, aflatoxin would be an example of that. Having said that, I think we have also evidence for things that we believe are exogenous, we believe might be related to diet, and this is purely based on epidemiology, and we don't see transcription or strand bias. So it is foreseeable that there will be something that won't have a bulky adduct, or it will be just damaging. DNA in a different way. But again, we don't have an example for that. It's just speculation.
Because it's also we have this very distinct different types of cancers in different regions of the world.
Yes.
In Sweden, we are suffering from prostate and breast cancer.
That's true. Yes.
In other countries, there's suffering from stomach cancer or from liver cancer or some other cancers. And then I wonder, what can you really explain this? From exposures causing mutations, or alternatively, could it be explained by some cell cooperation or some different environmental factors or some other factors that then cooperate to make some specific cancers. It may be not mutations. You might have environmental agents affecting the cooperativity or other
epigenetic factors also. Yeah, I think it's probably a combination of all of the above, to be honest.
Because there's been a lot of focus on, rightly, on mutations, but also could we have environmental then insults affecting how other ways of developing cancer, like the cooperation? It's an early question, I guess. We're very early in the science there.
So I think so personally, every time I look at stomach cancer, liver cancer, something of the GI tract, I always think this must be environmental. This must be something that people were consuming. And I usually think about somatic mutations. But you're right, it could be cooperation, it could be epigenetics, it could be modifying the tumor microenvironment. But usually when I think about, again, stomach cancer, there being very, very high prevalence of stomach cancer in China, I start thinking about something happening in the food. Now you mentioned...
Or Helicobacter pylori.
Yeah, that's our bacteria, but it's something seems to be related to the environment.
Yeah.
Now when it comes to prostate cancer and breast cancer, I find it harder to start thinking that there's something related to exogenous factors. And it very may well be, but I do wonder whether there is no lower risk predisposition factors in certain populations.
We've seen these cancers, the prevalence has increased over the last few decades. So if we have an increase in these cancers, so we have an increase, 300% of prostate and breast cancer, it's a lot of increase in these cancers. So how would you explain that? In the Richard Doll in Oxford, he found that of course there was an increase in lung cancers, and he was able to connect that increase in lung cancers with smoking.
Yes.
So how, in your view, do we see this increase in breast, prostate, ovarian, testicular cancer? What is your sort of view on this?
If I had to guess, I was going to say...
Is it environmental agents, or is it just random, or is it some new corporation?
I tend to think it must be something in the environment. I don't think there's anything innately different about us as people that's causing us to have these more cancers. I mean, I think with respect to the hormone-driven cancers, there's been work looking at the different chemicals in plastics and the hormones that we feed to our animals, and I think there's probably a lot of different things that we can look at as far as what's contributing. But,
and going to this plastic, and can you tell us a little bit about what you think about the plastics?
I honestly don't know enough about it to comment intelligently, but I think it's...
But I think you're very right, and we've seen that a lot of the additives to plastics and also plastics is hormone mimicking agents, and that we have flame retardants, for instance, that are very stable, they're very stable and they accumulate in the environment, and they're also hormone mimicking, and this is a concern. I think it's a concern that we use these ones and we don't really know what these compounds are doing. And so to my view, I had Thomas Lindahl, and he was saying that we don't live in a sea of mutagens, but I'm not so sure. There is a lot of plastics. We see these increased rates of cancer. We don't control what we put into even clothing, or things that we put into things that we wear on our body every day. These very hydrophobic compounds that easily pass through the skin. I'm not sure. I think there's a call for more research, but it is. I do believe, as you say, that it's probably environmental factors. But going then in preventing cancer, because we can treat cancer. That's something that we think we should do, but which is important, of course. But even better would be to prevent cancer. So how would, well, if I start with Allison first, how do you think that your work could lead to a prevention or to of cancer? Or
I don't know about tumor cooperation or tumor cell cooperation leading to prevention. But one of the things that I'm really excited about is this field of immunology in cancer, and this idea that you might be able to develop a vaccine or immunologic therapy you could administer to people to act as a preventative. I think that's really, really, really exciting work. And then, of course, with your work identifying different exposures, you could obviously eliminate those exposures and try and prevent cancer that way. So.
So now you answered Ludmil's
I'm
question.
sorry.
That's fine. That's good.
But going back to the immune field, and it's been a explosion in the cancer area on immune oncology, because it's particular when it comes to treatment. But also when it comes to the prevention, I think the cell cooperation, this is something that the immunologists have been working on for a long, long time.
No, that's true. That's true. Yeah.
So can you say, what is your thoughts? Is there cooperation between also the immune cells? You didn't look for cooperation between immune cells and the cancer, but the cancer need to evade the immune system, right?
No, absolutely. I think all of those things are acting in concert. So we have interactions occurring between the tumor cells. We have interactions occurring between the tumor cells and the immune cells. Interactions occurring between the tumor cells and the stromal cells, and it's all incorporating. And we have the invasion of the blood vasculature, and it's all acting as its own tissue, its own ecosystem. And I don't know that you can separate out, or we can try to separate out each individual component, but at the end of the day, it's all part of a bigger picture that's all working together, I think. I mean, they're going to need a multifaceted approach. Instead of trying to make a target against a somatic mutation or against a cooperative interaction between epithelial cancer cells or an immune approach, maybe take a similar approach to how we look at HIV, where we treat it from all different directions and try and sort of bombard the tumors from all of these different aspects. I think that might be our best shot.
Okay. You were also talking about the tumor microenvironment, which is also something that I think many people, when reading your work, come across and think that, well, this is, now you see cooperation between cancer cells. But previously we've known that there's been a lot of cooperation between the soil,
Right.
the tumor.
Yeah, exactly. And I think that's really interesting. And one of the things that's fascinating me recently is this idea about how that really influences metastasis. And if we have cooperation between different populations of cells, could that influence, for instance, metastatic tropism? We know in cancers that different or specific cancers tend to metastasize to specific places, and it's different for different kinds of cancers. And could it be sort of a honing mechanism almost that, with this seed and soil theory of metastasis, but we don't really know what makes the soil of one organ more favorable than another, and could it be because we have some cooperative interaction, or that organ can provide a specific signal that the tumor cells are seeking. And if we know that and can identify that, can we provide drugs that quelch that, and then we can prevent those metastatic events potentially.
This comes to me a little bit, what is your what is your next beef? Where do you find that the unanswered questions are that you would like to go for in the future now?
So this idea about metastasis is something that is really very interesting to me right now, and I think that's the direction that I would like to go next.
So how will you approach that problem?
That's a great question. I think we have some pretty powerful models being able to induce gene expression, for instance, in tissue-specific manners. And could it be possible to have, for instance, in the model that we worked in with overexpression of Wnt, express Wnt in a different organ besides the mammary gland, maybe inject tail vein injections or something like that into mice, and seeing do we see tumors arise in that extra that organ that we're expressing that Wnt.
So they would basically provide the fuel for the tumor in that area.
Yeah, and would it hone to that particular?
But I'm really curious about this cooperation thing.
Okay.
And you know what? Why do they? Why? It's very difficult to ask why, but obviously they want to survive. There's increased fitness by them cooperating. But is there something that we can learn from the evolution and the ecology field to sort of help us understand how this works, and how does it influence the tumor heterogeneity? Because one of the real key issues is the tumor heterogeneity. So to my mind, when you say that the cancer cells are cooperating, then you would have, if you have two cell types and they are cooperating, well, it should be fairly, if you need the two of them, they would grow out together, and in a sense, and then form a fairly homogeneous tumor. But this is not what we see.
That's right, yeah.
So we see a very heterogeneous tumor. Do you think that there is the similar cell cooperation in the different heterogeneous parts of the tumor?
That's an interesting idea. I don't know the answer to that, to be honest. I think we're just starting to really explore this idea. I don't know that all tumors have this cooperativity component to them. Certainly, I think a lot of tumors, particularly hormone-driven tumors, maybe the more well-differentiated hormone-driven tumors, could potentially have a cooperative component to them. But as far as all of the different subclonal populations, I don't know. I don't know the answer to that.
And there's another thing, thank you. And there's another thing also that I'm thinking. How do you think that this could be influencing other diseases other than cancer? Do you think that there's cooperation in other diseases other than cancer?
Now that's a really interesting idea. I haven't even thought about that at all. I'm sure it probably is, but I don't know that I could comment on that.
Okay.
So I'm sorry.
Do you think? Then I am also fascinated because you also said earlier about an aging phenotype that you see in one of your signatures, the signature 1, and this comes also to why we age, and if you see an aging signature, and it's caused by this deamination of cytosine to uracil, basically, or C to T transition. At least we think it's deamination of cytosine.
Yes.
Or you think it's something
Well,
else?
just to make a note, actually we see two signatures that are associating with age, and one of them, I think you're right. I think most likely the hypothesis there is deamination.
So why did you call them 1A and B? Because like you say, everyone is got a number, but there's one that calls 1A and B.
So when we did that, so actually in the very original paper when I did the analysis, we called them signature 1A and 1B, and we said from some cancer types we don't have the power, we don't have enough samples and or mutations to extract them. now we do have that power, and actually signature 1b have separated in two signatures, signature 1a, which we call signature 1 now, and signature 5. So both of those, signature 1 and 5, and it gets a lot of numbers now, but both of them start associating with age of diagnosis in different cancer types, presumably reflecting accumulation in normal cells.
So then you have the question, you have the answer to why do we age? So why do we age? Yeah, tell us.
I think, so obviously this is purely association with age, and I think what we are seeing with association with age is the damages that get converted to mutations. I don't think we're seeing the damages that are persisting on the DNA. And I think obviously, personally, if I had to guess, I think we age because there's a lot of damages that are accumulating in our cells and that are not being repaired.
So, so this is, you know, I'll challenge you a little bit on the aging. So do we age because of mutations?
That's the view I would like to that I would like to favor. I think there is other alternative views. So I would like. I'm sorry. I should rephrase that. I would like to say that we probably age because of DNA damages, not because of mutations. But I think there is another point of view that there is a predetermined clock in our body, and that eventually turns on, and you see women going into the menopause at the same age or similar ages. But personally, if I had to guess, I think we age because of damages, and I don't think somatic mutations play such an important role in aging.
So I agree with you there. So that's why I challenged you, because I'm not so convinced that it's the mutations that makes us age.
I think there are too few.
Yeah, so there's too few. So then the question comes, why do we age? And then when you talked about damage to DNA, and so why would the damage to DNA cause aging? What would be how do you lose sort of fitness? Because we age also because we lose fitness. You're clearly very
I
fit.
see.
And,
Thank you.
but in time, fitness will go down. And
I think that,
so what is about the fitness of the DNA? Is the fitness of the DNA going down in aging cells?
I think the fitness of the DNA is going down in aging cells. I think a lot of those cells are going into senescence, and I think that's why we're seeing the phenotypical differences between, you know, a 20-year-old and a 50-year-old. Question. Obviously, there is an interesting question whether we can go and.
You say it's DNA damage. So what type of damage is this, or you know, why would the DNA damage cause senescence?
I think just cells can either cannot replicate, or during the replication there is something that triggers a mechanism in the cells.
Yeah,
dies.
damage signaling.
Yeah, damage signaling. What damage causes it? I don't know. I think there is a lot of hypotheses with the mitochondria and reactive oxygen species. This is an interesting thing considering the mitochondria is outside of the nucleus. So how does this oxygen species go to the nucleus and damage it?
So I asked Thomas Lindahl this question, and he said it doesn't. And he said that there are some studies from the early days that the oxygen levels in the nucleus is extremely low.
Okay.
And he said it's water.
This is
it's you.
Yes.
But I also, because we didn't see that. Now you talk about oxygen and reactive oxygen species, but you don't report of any how do you get the C to T transitions from oxidative damage?
I think so. Yes, you have the C to T transitions, but I think when we look at that aging mutation or signature, it has other components, and some of them are C2A component, and I think some of them may be reactive oxygen species or may not be. It will be interesting to explore that. It's what you said about the water. It's an interesting thing. It's slightly depressing if it's water, because it's very little you can do with the water in the cell. But it's So I think it's obviously when it comes to oxidative damage, there's so many different types of oxidative damage, and I don't think we have explored them enough. So I think that would be a very interesting thing to look at in regards to both cancer and in regards to aging.
So I agree with you. This is probably important, but and then maybe there is not so much we can do about that. But it's at least if we can understand it.
Yes.
But I also want to challenge you. Is it only damage to DNA? You have other macromolecules in the cell.
So that's again, I think that's very interesting. Especially, I was very fascinated when I was thinking about bacteria. How does bacteria age? And then I realized actually damages to the protein, and when the bacteria divides, the damage to the copy that has more damaged protein is the one that's considered older. So it's probably we have these macromolecules that are very, very damaged. I think right now we are looking at the smoking gun with sequencing, because we can sequence everything. We can look at damages on large scale on DNA. I don't think we have that capability with proteins to do large scale examination of damages in proteins. If we, or maybe we do. But if we could, I think that would be extremely exciting.
So another question then, why do you think that the mutational signatures that you see are originated from damage to DNA? It could be damage to proteins.
It very well could be.
Generates the mutational signatures that you haven't understood.
Yes. So I think you're right. I think we have examples of other effects other than damage. So we think we have an examples of polymerases just being very dirty and not working. It is possible that there's damaged proteins that go and actually mutate DNA, or that inhibit some part of the replication process and result in mutations. It will be interesting to explore. I'm not sure how we can explore that, but it will be very much an interesting thing to check.
So when I also talked with Thomas, he said that Lindahl, he said that there are a lot of molecules in the cell that damage DNA. You have formaldehyde, and you have a lot of things that are produced endogenously, and he thinks that not all of them are found. And he also say that there are repair pathways and enzymes that we probably don't know of. So do you think that we have uncovered the majority of the sort of the damage that you see are generating? Do you have a good feeling what's happening in the large, if you take the bigger picture, there may be some details, of course, that we don't know. But in the bigger picture, do you think that you have a good view on what is causing the mutations in the skin cancer and the...
So what I think is that that's an interesting. The different chemicals is a very interesting point. But what I think is that we have found the main. So we have looked at quite a lot of cancer patients and cancer cells, and I think we have found the main processes or the main mutational signatures that cause mutation in human cancer. Now, what I suspect is happening is that a lot of them are combinations of different damages that we just don't have the power, or we may never have the power to disintegrate. So that signature 1 that associates with H, I mean, it could be a combination of deamination, endogenous alkylation, reactive oxygen species, but just because they're so similar between people, or the differences are minimal, we will never be able to see that. So I think from a mutation perspective, I think we are at least very close to mapping the main processes that cause mutations, the main signatures that cause mutations in cancer. But from DNA damage perspective and what actually results to this mutation, I think we're just starting that journey.
So now, asking, you got the question yesterday, what are you doing 5 or 10 years from now, and what did you say?
Well, what I said is that I cannot imagine myself doing anything else other than science. Yeah. I cannot imagine myself.
But then the question is that I would like to ask you.
Okay.
So what is your thoughts? What are you aiming for next? What is your lab? You have a very small lab that you've now started. What are you focusing on? What are you most interested in?
So as of right now, the thing I'm trying to explore, the thing I'm attempting to explore is epidemiologically different places. So as you were saying, Sweden has certain types of cancer, prostate and breast, that are elevated. In Native Americans, we have gallbladder and liver cancer. In the Czech Republic, we have renal cancer. So what I want to look at this in the extremes, the places that have the most cancer and of a type, so the highest age-adjusted prevalence for different cancer type, and contrast them either through time, if that has been increasing, and Japan is an example where colorectal cancer has been increasing for the last 40 years, so if we can get samples through time, or contrast them with other regions in the world or in a country that have very low risk for cancer. So yes, I think for the next 5 to 10 years, that will be the main focus of my research, trying to understand what causes these differences in cancer prevalence rate through genomics and through large-scale analysis.
Another angle on what you have done is also because therapeutics is very important. So how do you see that the mutational signatures that you have identified could be relevant for treatment?
I think that's a very exciting topic, and I think that very much actually comes close to your research of looking at and actually attacking failure of repair mechanisms. So we have a number of mutational signatures which tell us that certain repair mechanism in the cancer has failed, and I think we can start targeting. We can use them. We can use mutational signatures as an indicator for targeted therapy. So an example for that we recently did was failure of DNA double-strand break repair by homologous recombination. Obviously, it's associated with BRCA1 and BRCA2 mutations, but we see a lot more samples that have a specific pattern of mutations, and we believe that these samples will also be susceptible to PARP inhibitors.
But then you have also genetic reversion, heterogeneity.
That's true.
So how do you... This could be an ancient event that occurred many years ago, and then the cancer has evolved from there.
Could be an early event.
How do you separate the two? The kind of the actual mutations happening now, the ongoing mutational processes, or the old ones?
So I think in regards to treating a person, it will be so experimentally doing it for a few. I think one can do that. One can take cells and then do single clones and grow them and sequence them and see whether the process continues to accumulate. I don't think that's practical if you want to do it for a patient. You're very much right. I mean, we're seeing things that are, we're looking at an archaeological record. So this event may have occurred 10 years ago or 6 months ago, and it may not be ongoing. And we don't know. And also it could be that there is, even if it's ongoing, there could be a subclone that's completely different. So you're going to kill them, a big chunk of the cancer, but then you're going to have that resistance. And I think, echoing what Allison said, I think looking at combination therapy, I think looking at cocktails, very similar to what the HIV people were doing, I think that will be a game changer. And I think also the immunotherapies that are coming forward, I think there will be also quite influential.
So going back to you, Allison, then about, how do you see your work is relevant for treating cancer?
Yeah, so like I alluded to before, I think if there are cooperative interactions occurring between subclonal populations, we might be able to develop therapies, pharmacologic or otherwise, that
prevent
sort of
the cooperation.
Prevent the cooperation or interfere with the cooperation. Specifically where I see my career going, I'm a physician scientist. I'm going to be continuing my training in pathology, and I think there's a lot of opportunity for pathologists to sort of maybe bridge the gap between your end and my end, to take the very exciting things we're doing with cancer genome sequencing, and then be able to correlate that back with the actual histology of the tumors and try and see, are there subclonal populations? Can we dissect it out a little bit more to try and determine whether or not, the mutations we're seeing are global throughout the tumor, little pockets.
And I think this is such a nice path to take. We have looked too little on the actual, to look down the tumor is so important. And we've spent so many years on molecular biology and molecular cell biology without actually looking what's going on.
And actually, the pathologists would be able to have told you a long time ago that we know there's different cell populations. We see that. That's very common. So being able to correlate that back with the molecular studies, I think it's going to be a very exciting time.
So I think also your attention to detail here, when you're looking at these samples and being access to, how do you see that? Because what you need is to have, a lot of people have looked down to the cancer, but what you need is to have a different pair of goggles, basically, new technology, to be able to investigate this in another way. So where do you see that is coming?
So, off the top of my head, building on the work that we did in our lab, being able to take a solid tumor and actually analyzing it the way that we analyze a liquid tumor, being able to use flow cytometric technology and separate them into the different populations and identify the different mutations that they have to classify the tumors further. So taking a solid tumor, turning it into a single cell suspension, and then being able to isolate it. There's really exciting work being done now, being able to use in situ techniques where you can actually label intracellular mutations or protein changes to try and pull those
visualize
different subpopulations out. I think it's going to be really exciting, and we're going to be able to hopefully take each person's individual tumor and my goal or my dream, be able to solve it like it's a puzzle and find out what's really driving each person's tumor. Whether or not we'll be able to have keep up with the pharmacologic, the treatments to then address each person's tumor is going to be a whole nother challenge, but at least in dissecting what's driving tumor growth, I think that's going to be our first important step.
So we soon need to round up here, but I want to ask you also, for you, what has this? You have now received the Science SciLifeLab Prize, and what would you say is for you, Allison? How do you think that this can be useful for you?
Well, certainly, I'm so excited for just the exposure that this prize is given to my research, and I really hope that it inspires people to get excited about this topic that I'm so excited about. Other people to maybe be inspired to throw their hat in the ring and continue the discussion. Hopefully, people will be excited about funding this type of research, and so being able to make connections with all of the fabulous scientists here
and
in
also
Sweden
progressing
and around
careers.
the world.
I think this is something we want to do, and
absolutely
ensure that you, the prized minds, you are the prized minds here in Sweden, and that you are very successful in the future.
Yeah, well, it's truly an honor.
And for you, Ludmil, do you think that this, what would you say in a few words, just what does this prize mean to you?
I think this prize was first of all, it was really, amazing. And it was a fantastic experience coming to Stockholm and meeting with the other prize winners, as well as meeting with so many scientists in different fields. And I think very much, again, echoing what Allison was saying, I think this is going to be a very good opportunity to make our research more public, more well known, as well as hopefully allow us to get better funding or better score on our funding applications and make us more competitive. And as you said, advance our careers. So it has been, I think, we both had a great time here, so it has been quite an exciting.
So thank you very much, and again, congratulations to your fantastic work and your prizes. So thanks for coming.
Thank you so much. Thank you for having us.
Thank you.